Okay, I think we'll get started. And I want to welcome everyone to this IARS virtual education session on the anesthetic effects on neuroplasticity in the developing brain, clinical aspects, and research insights. My name's Bev Orser, and I'm the chair of the Department of Anesthesiology and Pain Medicine at the University of Toronto. And I also have the honor of serving on the IARS Board of Trustees. And today we have 4 outstanding world-class scientists and investigators, and I'm very excited about the session. But before I introduce our speakers, there's a few housekeeping details. The first is that this webinar has no commercial support and that CME credits are available to IARS members. I hope you'll consider joining us in April of 2023 for the IARS annual meeting, which will be held in Denver, Colorado. And just a reminder that starting June 1, the website will be open for session proposals. And so the meetings are built on a competitive basis of the sessions we get from our community. So please consider making your ideas about a symposium a reality by submitting through the submission process. The 2020 annual meeting recordings are still available for your viewing if you go to the IARS website. And after the seminar, you'll be able to continue to engage our speakers through the IARS Doc Matters community. So we can continue some of our conversations we start today. A recording of this webinar will be available to IARS members by the end of the week. And during the presentations, I encourage you to submit your questions to the question and answer option, which you can find at the bottom of the page. So question and answers rather than chat. After the session, you will receive an email about CME credentials, and information will come to you via email. And I think that's about it. Now it's my tremendous pleasure to introduce our 4 speakers. And the plan for today is that I will do the introductions, and then we'll have our 4 talks presented, and then we'll open up to question and answers. So the first speaker is Laszlo Vutskits, who is located in Geneva. He received his medical degree from Semmelweis University in Budapest, Hungary, and then completed a PhD in developmental neurobiology from the University of Geneva Medical School. He then undertook residency training in anesthesia in the Department of Anesthesiology, Pharmacology, and Intensive Care at the University Hospital of Geneva. Following his residency, he turned his interest to pediatric anesthesia and undertook subspecialty clinical training, but continued his basic science and translational work. And he's been a principal investigator with the research group in the department at Geneva Medical School. His principal clinical interest is post-op behavioral and cognitive outcomes in children. Now he holds many senior leadership positions, including being president of the European Society for Pediatric Anesthesiology. He is an executive editor of anesthesiology and a section editor of pediatric anesthesia. And he lets us know it's 10 p.m. where he is this evening. Our second speaker is Mary Ellen McCann, who's a pediatric anesthesiologist at Boston Children's Hospital, where she has worked for 30 years. She's an associate professor of anesthesia at Harvard Medical School. And her clinical research, which all of us are probably familiar with, has focused on the safety of general anesthesia and sedatives with regard to their actions on neurodevelopmental outcomes in infants and toddlers. And she's collaborated and led several multinational outcome studies, including the GAS trial, which was a very important study because it determined that short to medium length general anesthetic exposure early in life does not increase the risk of poor neurocognitive development compared to awake regional anesthesia exposure. She's very interested in the importance of conducting general anesthesia in very young infants and in very young interest. Also, preserving brain function is difficult in young children who may be inadvertently hypotensive, hypercarbic during routine surgical procedures. So she's been very interested in the importance of maintaining homeostasis during surgery in this group of patients. Her interest in pediatric neuroanesthesia extends to administrative and leadership roles as the chief of pediatric anesthesia for spine surgery at Boston Children's Hospital. And she's interested in outcomes for scoliosis surgery and developing strategies to reduce blood loss, reduce awareness, and effective wake-up tests for neuromonitoring techniques in these patients. So our third speaker is Guang Yang, who is an associate professor in the Department of Anesthesiology at Columbia in New York. And her research focuses on understanding how anesthesia and sleep and the immune system regulate the brain during development and function. She uses in vivo two-photon microscopy to monitor fluorescently labeled neurons in living mouse cortex. And her studies indicate that common anesthetic drugs affect synaptic development and plasticity in mice in age and dose-dependent manners. Multiple but not single exposures to anesthetics in the neonatal brain impairs learning and learning-dependent neuronal network activity later in life. And our final presenter will be Zhongkong Zi, who we refer to as Zi. He's well known to many of you, I'm sure. He graduated from Zhuhou Medical School at the university in China in 1985, and then obtained a PhD in pharmacology at Wayne State University in Detroit, Michigan. He then moved to medical school in Boston in 1994 and undertook postdoc research training in both clinical and preclinical studies. And in 1997, he returned to anesthesiology residency at Mass General and the Harvard Medical School. Following completion of his clinical training, he decided to become a physician scientist and continue his neuroscience research at Mass General. His work includes investigations of the neurogenesis of Alzheimer's disease, post-op neurocognitive disorders, delirium, focusing primarily on determining the roles of perioperative factors such as anesthetics, surgery, pain, sleep disturbance in the neuropathogenesis of these disorders. His work has been consistently supported by NIH grants and really represents the broad spectrum from basic science translational work to clinical trials at all levels. He's the Henry Knowles Beecher Professor of Anesthesia at the Harvard University and physician anesthesiologist at Mass General. He is the Director of Basic Science Research and the Director of Geriatric Anesthesia Research Unit in the Department of Anesthesia, Critical Care, and Pain Medicine at Massachusetts General Hospital and Harvard Medical School. So we really have an exceptionally distinguished group of presenters today. Thank you all for joining us. There are over 200 registrants from 37 countries. So this is a really wonderful opportunity for our community to get together and again, listen to some leaders in the field. So Kristen, can I ask you please to start the videos?
Good morning, everyone. My name is Laszlo Vutskits. I'm head of pediatric anesthesia at the University Hospital of Geneva, and I'm truly honored to be part of this IARS webinar on neuroplasticity in the developing brain. So in this webinar, I was asked to talk about what anesthesiologists should know about neuroplasticity during brain development. And obviously, given the time restraints, I will not be able to give a comprehensive view on neuroplasticity. But what I would like to do is to focus on some of the fundamentals which I think should be known by anesthesiologists, and these fundamentals could help to better shape future experiments both in the translational and in the clinical arena. So the first thing I'd like to do is to draw this distinction between neurotoxicity and neuroplasticity, because I think it's way more than just a matter of terminology. So toxicity comes from the Greek word toxikon, which was the substrate that the Greeks put to their arrow to kill the enemy. So toxicity depicts the degree to which a substance can harm, and it's often expressed as lethal dose 50 or lethal concentration 50. In contrast, plasticity depicts something that refers to the capacity, the capability of something or someone of being shaped due after or due to an external stimuli most of the time. So the capacity of adaptation. So in this context, neurotoxicity we could define as the degree to which a substance can cause physical brain damage, that is cell death. And plasticity, it's more neuroplasticity, it's more refers to the changes and the adaptation in neural pathways and synapses due to a change in environment and change in homeostasis. In terms of neuroplasticity, there are obviously several underlying mechanisms guiding this. Obviously, that we can have altered neurogenesis, programmed cell death, apoptosis is part of the mechanism that can drive neuroplasticity. But most importantly, I would like to focus on synaptic plasticity, which corresponds to the formation or the elimination of synapses or the remodeling of the shape of the synapses and their properties. And also, I'd like to draw the attention to the concept of maladaptive neuroplasticity when this when the normally, usually physiological neuroplasticity overshoots and results in a disease states, in a malfunction of neuronal networks. Now, I also like to emphasize that this maladaptive neuroplasticity is not neurotoxicity, meaning you may not obligatorily see a physical damage for that you could detect neuroplasticity. Maladaptive neuroplasticity, it can be in very slight changes in receptor compositions, for example, in synapses, or just a very slight increase or decrease in synapse number. Second issue I'd like to draw the attention to is that general anesthetics are powerful modulators of synaptic plasticity. Here I show you a schematic view of the synapse, and you have a presynaptic neuron, a postsynaptic neuron, with neurotransmitters being released and linked to the postsynaptic receptors. And now anesthetics can alter these physiological equilibrium in several ways. They are obviously very often allosteric modulators of the postsynaptic receptors, but they can also influence presynaptic receptors, synaptic release, and recapture. So with all this, the exposure to anesthetics obviously can alter and does alter synaptic transmission. The third concept I like to show is that during brain development, early life anesthesia exposure is particularly interesting because there is a period which extends from the last trimester of pregnancy up to the first 2 years or nearly up to adolescence in postnatal life. But this brain growth spurt, which is around birth, it's really characterized by synaptogenesis and network development. And during this period, there are several time windows, which we can call critical periods, where development has some particular aspects. I will come back later on to this. And what you have to realize that both GABA and glutamate are here from very early stages of and their functional receptors as well from early stages of development. And we know that GABAergic and glutamatergic transmission plays a major role during the critical periods of synaptogenesis and network development. Just to extend this critical period notion a bit further, I think you all know Dr. Konrad Lorenz, who was working with incubator-hatched gray-leg geese, but he found that when he showed himself up to these geese after hatching in a very well-specified time windows, neither before nor after this time window, these geese considered him as their mother all life long. And since these early investigations, there are a lot of studies that suggest that there are these time windows when the neural circuitry is very sensitive to external stimuli, and indeed the development of neural circuitry is dependent on these instructive signals. And we know that the timing and the duration of GABAergic and glutamatergic signaling is very important during this period, and even short-term interference can induce long-term consequences. So in this context, if we consider schematically that the inhibition-excitation balance during brain development is mainly driven by GABAergic and glutamatergic neurotransmission, if with anesthetics we interfere with these physiological patterns of neurotransmission, we can push in one way or another the inhibition excitation bonds, and we can interfere with the activity-dependent shaping of neuronal networks. Now, in light of all this, I think it's not an overstatement to say that CNS development and anesthesia, exposing the developing central nervous system to anesthetics, is an excellent model to study synaptic plasticity. The questions we can ask: what's the impact of general anesthesia on neuronal network development? Most specifically, it's something I was particularly interested in over the past decades, how general anesthetics can influence synaptic development. And there are obviously several models we can use to study this, and speakers in this webinar will show you some of these models. You can use either in vivo or in vitro approaches. I will just focus myself on two different in vitro approaches. The first is to study excitatory synaptogenesis and the impact of anesthetics, where if we expose, you can see the animals at postnatal day 5 or postnatal day 10, either to a single bolus or a 6-hour-long propofol exposure. When you compare to control animals, you can see that this will result in the decrease in the number of the small dendritic spines, which are the postsynaptic sites of excitatory synaptic contacts and can be used to study excitatory synapses. So you can see that there is a decrease in the number of excitatory synaptic inputs after propofol exposure. On the other hand, which was quite the contrast, if at later stages of development you will expose these animals to anesthesia, it's not a decrease, but an increase in the number of excitatory synapses. And then later stages of life, even up to a 6-hour-long anesthesia exposure, doesn't really impair synaptic density, excitatory synaptic density. And this was not just propofol. We've shown with several other drugs very similar effects. The only two big classes of drugs where we didn't find such effects were the opioids and the alpha-2 agonists, meaning that anesthetics can have a developmental stage-dependent effects on synaptogenesis. And what's important, that even a single bolus exposure at postnatal during early life, and depending on what stage of this early life you expose to these drugs, can have lasting effects. Even 3 months after, you can detect effects of in terms of synaptic density that corresponds or that correlates with some behavioral dysfunction in adult animals after early life anesthesia exposure. Now, the majority of the work people were focusing on corresponds to study excitatory synaptogenesis, because these dendritic spines, as you can see here, those are the postsynaptic sites of excitatory glutamatergic inputs, are relatively easy to study. There are genetically modified animals. You can do post-hoc injections following in vivo exposure. But an interesting question is, and we were, in the past few years, we were quite interested in what's happening with inhibitory synaptogenesis. And to do this, we took advantage of Geoffrin. Geoffrin is a protein that anchors, it's like the PSD95 in excitatory synapses. It anchors the inhibitory receptors to the postsynaptic cytoskeleton. And we can label this Geoffrin, and we can electroporate into the precursors of pyramidal neurons. And to make a long story short, this approach allows us to detect, as you can see these tiny spots here, this approach allows us to detect the inhibitory inputs, the sites of inhibitory inputs onto pyramidal neurons during development. And as you can see that the number of inhibitory synaptic inputs increases during the first 20 days of postnatal life in rodents. And then we were looking into what's happening with anesthesia exposure. These are just some preliminary data I show you here. For example, here we exposed the animals for 6 hours to midazolam at 7 days, and this decreased the number of inhibitory synaptic inputs to pyramidal cells when we checked 5 days later. However, we didn't find any difference when we checked at postnatal day 15. So this is just an example that anesthetics can also impair inhibitory synaptogenesis, and there are several experimental models where we can study that too. So very briefly, I have 2 take-home messages. One is that I try to show you that general anesthetics are context-dependent modulators of neuroplasticity during CNS development. And in this context, I think it's very important to shift the focus from neurotoxicity, from developmental anesthesia neurotoxicity, to talk about and to think about developmental anesthesia and neuroplasticity. As it's not just semantic, because I think if we think about neuroplasticity, the way we will plan future mechanistic studies, either translational or clinical, I think the design may differ compared to when we want to look for neurotoxicity. And obviously, all these fundamentals I've shown you, the clinical significance in the absence of an as yet unidentified human phenotype remains unclear. However, I think that when we consider the impact, at least the theoretical impact that these drugs can have on neuroplasticity, search for a kind of a maladaptive plasticity in upcoming mechanistic studies, either human or translational, can be interesting. And also, we may not just think about neurotoxicity, but as it has already been shown in some psychiatric diseases where anesthetics can improve function, maybe there can also be some therapeutic potential, or they can improve at certain stage certain kind of plasticity and function when we expose the children at young age to anesthetics. Again, these are just pure speculations, and I will stop here and thank you for your attention.
Relationships that I need to discuss, and no off-label use of or investigation. Hello, thank you. My name is McCann, and I'd like to thank the host for inviting me to this meeting. Today, I'm going to talk about the GAS study and beyond, from clinical findings to mechanism studies. I have no financial relationships that I need to discuss, and no off-label use of or investigational use of any medications. So I'd like to start off with telling you a little bit about the GAS study. This was a multi-site randomized controlled trial comparing regional and general anesthesia on the effects of neurodevelopmental outcome in apnea in infants less than 60 postmenstrual weeks in age. We enrolled 722 patients in over 7 countries in 28 hospitals. Patients were randomized to either get awake regional with bupivacaine or general anesthesia with sevoflurane. We published our results in the Lancet and found that there was no evidence that just under 1 hour of sevoflurane anesthesia in infancy increased the risk of adverse neurodevelopmental outcome at either age 2 or 5 compared to awake regional. Of course, this study had limitations. The duration of exposure was just under 1 hour. The exposure was during a very narrow period of neurologic development, very early infancy. Most of the participants were male. And probably most importantly, testing at 5 years does not detect a lot of domains, such as the domain of executive function or social emotional skills, which develop later. There are many neurocognitive tests that have been published looking at anesthetic exposure at a young age, over 90. Here's just a sampling of the few or of some of those studies that show that there's increased risk with early exposure to anesthesia. All of these studies happen to be retrospective. There are studies, however, that show that there's no increased risk. Some of these are large retrospective database studies, and some of these are matched sibling studies, and the one and only prospective study, the GAS study, showed no increased risk. So we really don't know at this point whether exposure to anesthesia at a very young age predisposes patients to neurologic or neurocognitive difficulties later on. One of the difficulties studying this topic is that there's so many confounders. Probably the largest confounder is the underlying pathology of the patient. Patients that are not healthy to begin with may already be developing neurologic problems irrespective of whether they have surgery or anesthesia. Surgery is associated with neuroinflammation, which may lead to neurocognitive difficulties. It's certainly been shown to cause delirium in adult patients. And when we look at anesthesia, we need to worry about the possibility of anesthetic neurotoxicity, but we also need to worry about our management. Ruth Graham, a pediatric anesthesiologist at Manitoba, developed a model, which is what she calls the dual risk model or the diathesis stress model for pediatrics. And basically it is, if you have a resilient patient and you put them in a positive environment or a negative environment, you're likely to get a good result. If you put a vulnerable patient, however, in a negative environment, you're likely to get a poor result. And what would include a negative environment for these children? Well, perhaps prolonged surgery, anesthetic that is suboptimally managed or very neurotoxic, or prolonged hospitalization. Vulnerabilities could be congenital anomalies, prematurity, or sepsis. We all know it's very difficult to anesthetically manage infants. It's very easy to get hypotension, overventilate the patients, not give enough glucose, either give too much or too little oxygen, have the patients too warm or too cold, or extreme positioning of the neck, all predispose to postoperative problems and possibly neurocognitive issues down the line. And as a specialty, we've been kind of focused on the neurocognitive outcomes, i.e. the IQ outcomes. The outcome of interest for the GAS study was the Wechsler Preschool and Primary Scale of Intelligence, full scale IQ. But there's some evidence that, that it may be, the effects may be more behavioral than general intelligence. So this study was just published last year, and it is a systematic review and meta-analysis of the MASK study, the GAS study, and the PANDA study, and found that there was no difference in general IQ when you looked at the studies with exposure or non-exposed children, but there were significant difference in parental reports of behavior in children that were exposed to anesthesia at a young age. Recently, a group of experts in this field got together and published a paper, a white paper in anesthesiology, basically looking at unanswered questions and proposed paths forward. Our group suggested that we needed further randomized clinical trials where there is a follow-up study to the GAS study known as the T-REX trial that I'll talk about in a few minutes. We need more observational studies, especially studies looking at dose response curves. We need perhaps to be looking at more studies looking at prenatal exposures. And this is taken from the environmental literature that shows that early exposure prenatally to some environmental toxins can cause measurable differences in children. And we need, of course, some more non-human primate studies, primates being non-human primate studies being very close relatives to us. And we need to think about the use of intermediate outcomes or biomarkers. If we have to wait close to 30 years for a human to fully develop, we just don't have enough time to wait that long. So to go to the T-REX trial, which is a follow-up for the GAS study, this is a phase 3 RCT, multi-center, multi-country superiority trial. Its inclusion criteria include children under the age of 2 scheduled for surgery lasting at least 2.5 hours. So we're trying to get at that issue of prolonged anesthetic exposure. And basically it's high dose sevoflurane versus low dose sevoflurane with remifentanil and dexmedetomidine. And this study is over half enrolled at this point. Our outcome measures are neurocognitive testing and behavioral testing at age 3. So what might be some intermediate outcomes or biomarkers that we might want to be interested in? Well, endophenotypes refer to what is going on inside the body. And the easiest endophenotype to look at are imaging studies such as MRI studies. If we want to get at the mechanisms of injury, then we might need to figure out what biomarkers or what biomarkers are changed during anesthetic exposure. This can't be an inquiry in and of its solely on itself because there are limitations. Biomarkers need to be correlated with long-term outcomes of interest. It may be difficult to determine whether differences in biomarkers are due to anesthesia or other perioperative exposures, such as the surgical experience leading to neuroinflammation. We are involved in a biomarker study looking at tau and p-tau. Dr. Z and myself are conducting the study at Boston Children's and at Mass General. And basically, we are looking at baseline levels of these markers and then postoperative levels of these markers. And the children are exposed to sevoflurane. And we found that there is a definite increase in plasma tau as well as urine tau in several of the patients that we've already enrolled in this study. These patients come back a year later and get neurocognitive and neurobehavioral testing by way of the NIH toolbox. So to conclude, there is the evidence of pediatric anesthesia-induced neurotoxicity from early anesthetic exposure, from retrospective studies and even prospective studies is still equivocal. There's a faint signal that neurobehavioral outcomes are more effective than neurocognitive outcomes. Many different areas of research on this topic are warranted, and studies looking into endophenotypes and biomarkers are necessary to determine mechanisms. With that, I'd like to conclude and thank you for your attention. Bye-bye.
Hello, everyone. My name is Guang Yang, and I'm pleased to join this webinar from Columbia University Medical Center. Today, I will be talking about anesthesia effects on neuroplasticity in developing brain, with a focus on the role of brain circuits. I have no conflicts of interest. As we all know, neuronal activity plays a key role in the development and maintenance of neuronal circuits. In developing brain, the timing and the pattern of neuronal activity are fundamental for the proper formation and refinement of neural networks. Interference with this physiological pattern of neuronal activity during critical period of brain development can have long-term functional consequences. One prominent example comes from the developing visual system, where spontaneous and visually evoked activity drive the maturation of visual circuits, and the blockade of such activity can cause permanent vision loss. So in this context, it's not surprising many animal studies have consistently demonstrated the long-term impact of early life anesthesia exposure on behavior and cognitive function. This is because anesthetics are powerful modulators of neuronal activity. As shown here, one injection of propofol in young mice at a dose to induce a surgical plan of anesthesia causes a prolonged reduction of neuronal activity that persists into the anesthesia recovery period. This raises the possibility that anesthesia-induced prolonged suppression of neuronal activity may impair neural circuit development and thereby contribute to behavior and cognitive changes. Here we designed an experiment. Neonatal mice were administered 1 or 3 sessions of general anesthesia during postnatal days 7 to 11. Control mice received weekly injections. At 4 months of age, these mice were subjected to a series of assays at behavior, molecular, and cellular levels. In line with previous observations from my own lab as well as from many other labs, we found that multiple exposures of neonatal mice to general anesthesia induce a long-term behavior, cognitive, and motor impairments in adulthood. These functional deficits are associated with decreases in synaptic protein expression, decreases in excitatory neuron density, as well as decreases in synaptic density and plasticity in the cerebral cortex. And by contrast, a single exposure has no such effects. To further understand the functional deficits of neuronal circuits, after early life anesthesia exposure, we use the in vivo 2-photon imaging technique in combination with transgenic mice expressing genetically encoded calcium indicator to assess neuronal calcium activity in the cortex of awake behaving mice at 4 months after neonatal anesthesia exposure. So here, changes in calcium fluorescence serve as a good proxy for neuronal activity. It's known that in healthy mice, motor learning increases neuronal activity in the mouse motor cortex. So this video was collected from apical dendrites of pyramidal neurons in the motor cortex of mice running on a treadmill. As you can see, treadmill running is associated with an increase in synaptic and dendritic calcium activity. And this increase in activity has been shown to be learning dependent. Comparing mice with and without neonatal anesthesia exposure, we found the treadmill running induced pyramidal activity is much lower in anesthesia exposed mice. So this is also consistent with the reduction of motor learning performance in these mice. While pyramidal neurons represent more than 70% of all neurons within the cortical circuitry, there is also a large diversity of GABAergic interneurons there. It's well established different types of interneurons target specific domains of the principal neurons or other interneurons, providing precise spatial temporal control of excitatory and inhibitor outputs of the cortical circuits. To further understand the long-term impact of general anesthesia on neuronal circuits, we examine the activity of local inhibitor cells in the primary motor cortex. In the cerebral cortex, 3 major subtypes of interneurons, somatostatin expressing, Pavlovin expressing, and VIP expressing interneurons, are connected in such a way that SST and PV interneurons preferentially inhibit pyramidal neurons, and VIP interneurons directly inhibit both SST and PV interneurons. Our results showed that motor training-induced activity in VIP interneurons is significantly increased in propofol-treated mice as compared with vehicle-treated controls. Consistent with the connectivity scheme that VIP neurons inhibit SST and PV cells, we observed a marked reduction in SST and PV cell activity in mice with early life anesthesia. So our conclusion is, repeated but not a single exposure of neonatal mice to general anesthesia causes motor learning impairment in adulthood, which is accompanied by a reduction of pyramidal neural activity in the motor cortex. The activity of local inhibitor interneural networks is also altered. SST expressing and PV expressing interneurons are less active, whereas VIP expressing interneurons are increasingly activated when the mice are performing a motor learning task. Without going into details, we also found that using pharmacological approach to accelerate the recovery of neuronal activity in anesthetized mice, for example, by injecting non-competitive GABA-A receptor antagonist PTZ, all the AMPA receptor agonist CX546 can protect these mice against anesthesia-induced long-term functional consequences. So bringing these findings all together in a model, I believe anesthetic agents cause profound changes in brain activity, and alterations to brain activity during a critical period of neural circuit development, triggers long-term functional consequences. Ultimately, this leads to changes in behavior and cognitive function in adulthood. And one key to protect mice against the long-term effects of the early life anesthesia is to accelerate the recovery of physiological patterns of neuronal activity during anesthesia recovery period. Finally, I would like to thank the whole team that carried out this study, our collaborators and funding sources. With that, thank you, and I look forward to hearing your questions later.
Good afternoon. Thank you for the invitation. I'm very honored and happy to present you our research regarding anesthesia effect on neuroplasticity in developing brain, the role of tau protein and neuroinflammation. Here's my disclosure, and the research was supported by grant from NIH, Alzheimer's Association, and Q Alzheimer's Fund. And I provide consulting service to Baxter, Novartis, and Shanghai Nines and Cancer Hospital. However, the study reporting here have no any connection with any pharmaceutical companies or institutes. So the speaker has talked about very well regarding clinical studies. We all know that the clinical study may take a long time to conduct and analyze with confounding factors and other limitations. Therefore, there's a need to perform the animal research focus on mechanistic hypothesis, vulnerable window, less proactive anesthetic, and potential treatment and prevention. So in 2003, the pioneer work by Dr. Genovich and Tolovich showing that the cocktail of isoflurane, nitrous, and Versed can cause learning memory impairment in rodents and also the cell death. And in 2009, Dr. Sadamoto in Japan showing that 3% sevoflurane does not cause significant changes in terms of the blood gas and cerebral blood flow. So we also using the 3% sevoflurane. We're giving the young mice 2 hours daily on day 6, day 7, and day 8, or on day 6, day 8, day 10, or on day 6, day 9, day 12. So 3 times treatment can cause learning memory impairment at about 1 month after their birth. So we try to find out what's potential mechanism. And we know that many things can contribute to such learning memory impairment in young mice, including apoptosis, beta accumulation, tau protein phosphorylation, and neuroinflammation. So we're going to focus on tau protein for current studies. So tau protein is usually binding to microtubule inside neurons. When it becomes the hyperphosphorylated, it will detach from microtubular to form this thing called tangles. We first found that sevoflurane, 3% actually, can only cause learning memory impairment and brain tau protein phosphorylation at 3 days in day 6 by young mice, day 6, day 8, day 10, or day 6, day 7, day 8. Not in the adult mice, and also desflurane doesn't cause such effect. And the 1-day treatment of sevoflurane also does not cause such effect. So therefore, it is a drug-dependent, time-dependent, age-dependent tau protein phosphorylation and learning memory impairment in the young mice. We also found that sevoflurane can cause tau protein phosphorylation, causing increase in IL-6 generation, leading to mitochondrial dysfunction, synaptic loss, and learning memory impairment. So the question that how that happens, how tau protein phosphorylation can leading to generation of IL-6, and we hypothesize it's causing trafficking of tau, leading to this change. So here's the data. We're first showing that giving sevoflurane to young mice, we can observe the increase of tau in the blood of young mice. But desflurane, on the other hand, doesn't increase the tau in the blood. Last, we found sevoflurane can cause tau protein phosphorylation inside neurons, but again, not desflurane. We further found that sevoflurane can cause tau protein come out from neuron into extraneuronal culture medium for both tau and p-tau. But the LDL, which has similar molecular weight, doesn't come out, suggesting that the sevoflurane can increase the specific tau trafficking from inside neuron to outside neuron. And again, desflurane does not increase the tau come out from neuron. We further found that sevoflurane, but not desflurane, increase tau and p-tau in the extracellular vesicles. So here's also from sevoflurane, we see such increase in the p-tau in these EVs, but not desflurane again. And we also found sevoflurane increase tau inside microglia. This is a tau lockout microglia. We shouldn't see any tau. If we see tau, that means something is from external resource go inside the tau lockout microglia. We found if we treat it with conditional cell culture media, which containing tau, it can penetrate inside of tau lockout microglia. Same thing happened for the immunostaining, also for nano beam technology, showing that the tau and the p-tau can indeed go inside microglia. And over there, it can cause IL-6 generation. So if you're giving the neuron only with sevoflurane, no increase of IL-6. Microglia only, again, no such increase. If we add on both neuron and microglia together, sevoflurane can increase the IL-6 generation, suggesting we need both neuron and microglia to have generation of IL-6. So here is our potential mechanism. Sevoflurane can cause tau protein phosphorylation inside neuron, but not desflurane. When tau becomes phosphorylated, it should have just like an airline ticket. It can come out from neuron to outside of neuron. In outside of neuron, it can carry out by extracellular space, by extracellular vesicles to go inside to microglia, leading to generation of IL-6 inside microglia, causing linear memory impairment. Desflurane, because it doesn't cause tau protein phosphorylation, therefore it didn't cause tau come out from neuron to extracellular space, therefore no increase of IL-6 and no change of linear memory function in the mice. So here's a summary. Anesthesia, like sevoflurane, can induce a selective tau protein phosphorylation and cognitive impairment in mice with age, time, and drug dependent. Sevoflurane induced tau protein phosphorylations, which then causes IL-6 generation, leading to mitochondrial dysfunction, selective loss, and linear memory impairment in the young mice. But how the tau protein can lead to IL-6, because sevoflurane can cause tau trafficking from neuron to microglia through these cellular vesicles, leading to generation of IL-6. And much more research work are needed in studying anesthesia effect on neuroplasticity in developing brain. And I've thanked many people in my lab, our collaborator, and also the spokes from AIRG funding, our Department of Anesthesia at Mass General Hospital, and the Harry Beecher Professorship from Harvard University. And thank you for your time.
Wonderful. Well, thank you very much for 4 outstanding talks. And more importantly, thank you for the cutting edge research you are doing in efforts to keep the brains of young children safe. Wonderful. So I've tried to capture some of the questions from the chat, and I'll start with a question to Laszlo. Does plasticity change with moderate stimulus? Can you comment on the dose-dependent relationship between exposure and changes in synaptic plasticity?
Yes, so I mean, obviously it looks surprising, but if you consider that there are several experimental models, we couldn't show it here, but when you you can video these changes in synapse formation, and actually even under normal condition, it's relatively rapid. And obviously, when you work with neural activity, I'm not even sure that it's, I mean, with general anesthetics, in terms of plasticity, you don't really need to, it's not really a dose-dependent thing, or maybe it can be a dose-dependent thing, but even with tiny doses, when you modify neural activity, very quickly you can induce activity-dependent changes. Now, again, these are experimental models. Most of them are experimental models. And as Mary Ellen showed this, we do not yet have a clearly identified human phenotype. But for example, in terms of other moderate stimuli, for example, if you induce epilepsy in mice, or if you increase carbon dioxide levels, you can induce dendritic synapse elimination. And just we have some evidence in adults. For example, we've done a study with patients in major depressive disorders where we just expose them a single subanesthetic dose of ketamine just once. It's a half-life, effective half-life less than 2.5 hours. And then we've done several functional MRI imaging with the reward system. And up to several weeks, the reward system was rewired with a single dose of ketamine. So yes, it does exist also in humans. Again, in terms of small kids, in the absence of a clear phenotype, at least the way we are looking for it now, we don't know what it means, but it does exist.
Great, thank you, Laszlo. Mary Ellen, next question's for you. And you, building on this notion of imaging and biomarkers, can you share a glimpse into the future and your thoughts about what those biomarkers might be?
Well, I think Dr. Z's talk about tau and phosphorylated tau is very promising. And he's actually offered a mechanism by which there could be a cause for the neurocognitive differences. So I think that's very exciting. I personally am not too enthusiastic about some of these MRI studies. I don't know whether they're going to be granular enough to actually detect differences. But obviously that's something that people are looking at, and there are some retrospective studies that have looked at MRI changes in children who have had anesthetics. Again, they're so confounded by anesthetic management and the underlying diseases of the children that I'm personally not too, I'm not waiting with bated breath on those kind of studies, but it's something that we can't discount entirely.
Thank you.
You're welcome.
Guang, a question from you. Thank you for sharing your very sophisticated techniques and exciting data. Have you, using these methods, have you identified any strategies to mitigate or change the changes you're seeing using the in vivo confocal approach?
Oh yeah. So our previous study showed basically two strategies. The one is the counter the drug effect of anesthesia on brain activity. So that obviously you could use pharmacological approach, as I mentioned, to use some GABA receptor antagonist or the AMPA receptor agonist to help in that purpose. Basically, the idea is to quickly bring back the neuronal activity to the normal level after the end of the general anesthesia. And in our previous studies, we also showed for the animals, for mice, even after repeated exposure to general anesthesia, if you leave those mice in an enriched environment, so that's a natural way to bring more sensory stimulation to help the development of the nervous system. That could also counter the adverse effects of the early anesthesia exposure. So I think that could be, it has more translational insight. So in the context of the human beings, I think it probably is different from the animals. So naturally, we are going to have more enriched education environment. So that may have a more profound effect in terms of to help to promote the cognitive function development.
Thank you. Z, next question goes to you. And first of all, thank you for mapping out a really interesting signaling pathway involving glia, tau, cytokines. Very, very exciting work. I noticed that lithium mitigated some of the actions of tau. Can you comment on why you chose lithium and what the pathways and mechanisms might be?
Sure, that's a great question. We only try to prove that the trafficking of tau is totally dependent on the tau phosphorylation first inside neuron. Then it can come out from neuron to initiate such trafficking to microglia, causing neuroinflammation.
Great, thank you. Okay, we'll go back to Laszlo. This notion of sustained effects, and you comment about ketamine's actions and now these single exposures. Any common hubs in terms of reprogramming a genetic expression? Any insights about the upstream signals that are triggering the sustained change? It's a very big question, I know, but any insights that you might share? You're muted. Laszlo, you're muted.
Yes, thanks very much. Indeed, it's a very big chunk of question, and a big part of the neuroscience community is working on it. And even if you don't have a human phenotype, one of the things I wanted to show that anesthesia is really well placed as an experimental model to study this. And we know that there are several neurotrophic factors involved in it, and long-term modification, mTOR signaling pathways, BDNF pathways, just to cite a few. Zhongchong just showed some of the other aspects. So there are probably several up and downstream mechanisms involved in that. Basically the same players, those regulate neuroplasticity.
Thank you. And Mary Ellen, this is a question that's coming up a number of times. Dexmedetomidine is being widely used by our pediatric colleagues. And someone asks, is it time to change practice? And what do we know about dexmedetomidine in terms of its actions? Is it safe?
It seems to be very safe. If you look at the animal studies, it definitely seems to be associated with less animal neurotoxicity. It's got big drawbacks in clinical practice. Probably 60 to 70% of surgery done in young children is ambulatory. And if you're using a heavy dexmedetomidine technique, you can anticipate the kids will be pretty sedated postoperatively. So that's one of the issues. A certain percentage of kids will become hypotensive or bradycardic with it as well. So I don't know if it's quite time. I think everybody has to pay attention to the ongoing research for the next couple of years, because it may be that we're going to be increasing our use of dexmedetomidine and decreasing our use of sevoflurane. I mean, the one thing that I got from Z's talk, Dr. Z's talk, was that desflurane seemed much safer than sevoflurane. And we know of all the volatile agents that sevoflurane is the most associated with postoperative agitation. And we were talking about a phenotype. We don't know that postoperative agitation or postoperative delirium is a phenotype for neurocognitive or neurobehavioral deficits down the road, but it's possible. So it's just something to think about.
Thank you. I remember the recovery room nurses referring to it as screamoflurane, right?
Right.
That agitation.
Yes.
When you mentioned this notion of, are we allowed to go a minute over, Kristen? You get permission?
Yes, you can.
Okay, thank you. Guang, this notion of accelerated recovery, are you thinking of antagonists? And in fact, there's a question, what about the antagonists that the veterinarian colleagues use? They are available. Are you thinking antagonism or simply enriched environment?
So I would prefer to use the environment just naturally to bring up the physiological activity to counter the pharmacological effect.
Okay. And see, the last question goes to you. And Mary Ellen mentioned desflurane. Interesting. I think we're all using less desflurane for a variety of reasons. Environmental impact is certainly driving change in practice. Thoughts, is this, what distinguishes desflurane from SIBO mechanistically? And just your comment on its potential use as a cognitive sparing option.
Sure, great question. The answer is we don't know yet. You know, they're very structurally very similar, but have different effect. Our primary studies so far showing that potentially, only potentially, they may have different binding capacity to some enzyme, such as the ATP synthase. Using computer simulations, sevoflurane has kind of 3 binding sites with the enzyme, but that's only 1. Is that causing difference?
Dr. McCann, can you mute, please? Sorry.
Is that causing the difference? We don't know for sure yet, but that's a possibility. So they may have different interaction due to the energy level with different molecules.
Okay. Well, thank you to all of you on behalf of the over 100 participants who joined us this afternoon. Thank you for your talk, but more importantly, thank you for your incredible work. The IRS really appreciates you participating in this session, as do all the listeners. We'll look forward to seeing you in the future, hopefully at next year's IRS meeting. Thank you and good afternoon.
Bye-bye.
Thank you.
Bye. Bye, guys. We'll see you.